All right, so now that you guys have some background on the sex-influenced traits, we're going to switch gears and talk about sex-limited traits. And one of the classic cases is precocious puberty in males. So this trait is where males are actually going to be fully fertile by the age of 4 or 5. They go through puberty very, very early in life, and they do this because the there's a an autosomal dominant allele that we're going to call P, and that individuals with that allele undergo puberty at a very early age. So it's really an interesting case. So these individuals end up fully fertile, completely capable of reproducing at an early age. However, they tend to be very short in stature because with the production of testosterone, that will actually tie up growth factors in bone ossification. So they end up being very, very short. So the mechanism behind this trait for precocious puberty really lies in the gene or for the receptor of luteinizing hormone or LH. So LH stimulates testosterone, and the defective gene makes it so that the receptor um, that stimulates testosterone in the absence of luteinizing hormone. And so that ability to tie up the LH receptor basically allows testosterone to be produced, and this is really limited to males because in females you can't start puberty because you also need a second hormone for the interaction, and that hormone is follicle stimulating hormone. So that's why you see precocious puberty happening in males more so than in females, because there is a genetic defect in the receptor of luteinizing hormone. So, just looking at inheritance patterns, if we have two parents that we have one individual that went through precocious puberty and has a heterozygote genotype, and we cross that individual with with a homozygous recessive genotype, the F1 generation is gonna look like this. It's gonna be half of them are gonna have the heterozygous genotype, therefore they're going to go through precocious puberty because only one copy of the big P allele is needed for that phenotype to happen. If they're males. So here's where we really start to see differences between the sexes. So also in males, if we're going to have 50% of the individuals be homozygous recessive, and those individuals are going to go through normal puberty. However, on the female side, our ratios are going to be the same for the genotypic expression. So half of the genotypes are going to be heterozygous, but because those individuals are female, they're going to go through normal puberty because females require the second gene, follicle stimulating hormone, in the second hormone to actually start the pubertal process. And then lastly, 50% of the F1 generation of females are going to be homozygous recessive, but they're gonna also have the normal phenotype as well. So the parent generation is really, or when we actually do a reciprocal cross, where the male is now the homozygous recessive genotype in the female, and the parent generation is heterozygous, we end up with the same ratios. So it's very much a sex dependent issue. So the conclusion from this cross is that both males and females have the ability to pass on the sex limited trait, but it is expressed only in males. So switching gears again to cytoplasmic inheritance, and this concept is really quite interesting because you can actually see um, different genes being expressed because of the cytoplasm of the genes. So some genes that are found in the cytoplasm tend to be tied up in the mitochondrial organisms and the chloroplasts. And those two genomes are really pretty small. There's only 35 to 37 genes in the mitochondria or the chloroplasts. And that is really small compared to the genetic material that we see in the nucleus, where we're going to see about 35,000 genes on chromosomes. So, with mitochondrial inheritance, or cytoplasmic inheritance, specifically in the mitochondria, what ends up happening during the cell division process is that the mitochondria are going to segregate randomly during cell division, very much similar to the genes being randomly segregated during meiosis. But keep in mind, this is organisms with, organelles within the cytoplasm of a cell. So that random distribution really enables genetic material to not divide evenly. So sometimes you'll get more cytoplasmic inheritance in one cell versus another cell. So that's why we see a lot of the effects of the genes in the mitochondria and the organelle, or in the chloroplasts. Those offspring that inherit that from their mothers really display a phenotype that is very similar to their mother because of that cytoplasmic inheritance. This concept was demonstrated by Carl Correns in 1909, and he was looking at leaf color in a couple of different types of plants. And so what he ended up determining was that the leaf color was dictated by the maternal chloroplast DNA. So he did many, many different back crosses and test crosses to where he was looking at females or female seeds and pollinating with different phenotypes of male plants. So whether they had a lot of white leaves, no white leaves, or just a few spotted leaves. And the result really ended up being pretty consistent. And the conclusion was that pollen had no effect on the phenotype of the progeny. Switching gears just a little bit, so we just spent the last couple of slides talking about cytoplasmic inheritance, and now we're going to switch gears and talk about something similar but different, and that's genetic maternal effects. And a genetic maternal effect is where an offspring's phenotype is going to be determined not by its own genotype, but by the mother. So an example that we see this demonstrated in is snails. So snails are going to have a certain coil to their shell. If they have a dextral coil, then the coil is going to be to the right. If they have a sinistral coil, it's going to be to the left. And the bottom line is that the mother's genotype actually determines the offspring's phenotype. So looking at this example a little more closely, we really have two alleles that are present for this loci, S plus and S. S plus is the dominant right twist to the shell, and just an S is a recessive left twist to the cell. To the shell, sorry. So if we have two parent or in our parent generation, if we have a male that is dextral, we cross it with a female that is sinistral and homozygous recessive. In the F1 generation, we get the 100% heterozygous phenotypes, very similar to our dominant recessive mechanisms. However, if we allow interbreeding to take place between the F1 generation, our F2 generation looks very different because all of the phenotypes are going to be dextral in nature because that's what the mother was. But all of the phenotypes are going to be dextral in nature. But the genotypes are going to take on that classic 1:2:1 ratio where you see 25% having the homozygous S plus genotype, 50% having the heterozygous genotype, and 25% having the homozygous recessive genotype. So even though the genotypes are very textbook 1 to 2 to 1 for this relationship, the phenotypes are not. And so they are going to all 100% be dextral in nature because the mother's genotype is determining that phenotype. So there's a couple major differences between cytoplasmic inheritance and genetic maternal effect, just to make sure that you guys are clear on what these concepts are. Cytoplasmic inheritance is really dealing with the principle of genetic material and expression being transmitted through the mother's mitochondrial or cytoplasmic DNA. Whereas the genetic maternal effect is where the mother's genotype is actually determining the phenotype of the offspring. So similar concepts, but very different in mechanism. All right, and we are going to stop there for part B and pick up the rest of the lecture in the last part.